Good morning, gentlemen. Today is Monday, April the 6th, and today we are going to talk about concept 15.3, which deals with genes that are linked together on the same chromosome. All right. If two genes are found on the same chromosome, then we would expect that they would be inherited together. And so if mom has a certain combination of alleles, we would expect that combination to go to an egg. And if dad has a combination of alleles, we'd expect that combination to go to a certain sperm. All right. But linked genes tend to be inherited together because they are located near each other on the same chromosome. Now, each chromosome has hundreds or even thousands of genes, with of course, the exception of the Y chromosome in males. And we define genes located on that same chromosome as linked genes. Now, how does linkage actually affect inheritance? Well, Morgan, the guy who first worked with fruit flies in order to work out some of the chromosomal theory of inheritance, did some experiments to see how linkage affects inheritance of two separate characters. All right. For example, he used fruit flies, wild type over here. Whoops, I don't have my pen here. So the wild type fly is here, and this one is a double mutant. It is a black colored body and what we call vestigial wings, severely shortened and malformed wings. Now, when he bred these two individuals, he ended up, holy Christmas, with an entire generation of F1 that were all wild type flies, and they all had gray bodies and they all had normal style wings. He then performed a test cross between that F1 generation and another set of homozygous recessive black body vestigial winged flies. And in the F2 generation that he produced, he ended up with a number of different organisms. He ended up with wild type individuals, homozygous recessive individuals with both recessive traits, but he also ended up with gray-bodied vestigial winged flies and black-bodied normal winged flies. Now, this is the important portion here. All right. The predicted ratios, if the genes were found on different chromosomes, and remember that fruit flies have 4 chromosomes in their genome, then we would expect a 1:1:1:1 ratio here. But if the gene for body color and wing type were found on the same chromosome, in other words, if they were linked, we would expect to find a 1:1:0:0 ratio. In other words, none of these recombinant types that we see here. But in reality, what he found was that we ended up with almost a 3:3:1:1, actually, well, kind of almost a 3:3:1:1 ratio here. All right, and you can see the numbers that he received. And so that was a big moment. What's going on here? Well, Morgan realized that body color and wing size are usually inherited together, all right, 900 individuals, and in very specific combinations, which he referred to as the parental phenotypes, all right, because they looked like the parents. But he did note that they don't always assort, that they don't usually assort independently, and so therefore they were probably found on the same chromosome. However, there were also non-parental phenotypes, all right, non-parental phenotypes produced in the test cross. And we know now that this is the result of genetic recombination, all right, the production of offspring with combinations of traits that differ from either of the parents. So how exactly does this happen? Well, this occurs through a process we call genetic recombination. And we're going to study today what the effects of genetic recombination are on linkage. So how does this all work? All right, well, if two genes are unlinked and they start out on completely separate chromosomes, then we end up with completely independent assortment of those chromosomes. All right, and the genes that are on those chromosomes. So offspring that have a phenotype that matches one of the parents are called parental types. But offsprings with new combinations of traits are called recombinant types. Okay? And if two genes are found on different chromosomes, then you would expect to find a 50% recombination frequency. All right, and you might see that in the yellow round, yellow wrinkled, green round, and green wrinkled pea plants that Mendel studied. If we used a test cross and bred them all with a wrinkled homozygous offspring. Now, uh, what if the genes are indeed linked? Well, if they're linked, then you would expect that there would be no recombinant organisms at all, right? Because the two alleles, the two genes are found on the same chromosome. They should be inherited together always. But that didn't happen. Right? Go back to Morgan's experiment, 900 to 900 to 200 to 200 ratio that he found there. So Morgan observed that although some genes are linked, non-parental allele combinations, in other words, recombinations, are still produced. And he proposed that there's got to be a process that occasionally breaks the physical link between two genes that are found on the same chromosome. And we know today that that process is crossing over. And so in order to show this, if you examine the flies, this is the same experiment now. Here's the heterozygous dihybrid fly, test crossed with a homozygous recessive black body vestigial winged individual. Well, these guys are only going to produce gametes with the black body and vestigial wing genes, because even if there's crossing over, they're not going to produce any other combinations. But the hybrids can produce different combinations. And if you look very carefully at this portion, you can see the recombinant chromosomes that are located here. And therefore, there's not only two different sperm or eggs that can be produced, the parental types are shown here, but also the recombinant ones are shown here. All right. And so when you test cross these guys, you end up with 4 different phenotypes: gray body, normal wings; black body, vestigial wings; gray body, vestigial wings; and black body, normal wings. All right. And so we end up with 2 different kinds of parental phenotypes and 2 of recombinant phenotypes. All right. Go back to this and watch the PowerPoint presentation so that you can actually watch this animation. I can't do it in this video here. It takes too much time. Now, new combinations of alleles. Well, if you produce these recombinants, you end up with new combinations of alleles. And this is the variation that evolution by natural selection requires to do its kind of work. Now, how do we can use recombination data to figure out how far apart on a given chromosome 2 alleles actually are. We do this by constructing a genetic map or a linkage map here. We use the basic idea that the further apart 2 genes are on a given chromosome, the more likely it is that they'll be separated during crossing over events. And so we can use the frequency of this crossing over bit to build what we call a linkage map. Okay. Now, the distances between genes is currently expressed as what we call map units, where one map unit is equivalent to a 1% recombination frequency. And so, for example, if you did an experiment and you got a 28% recombination frequency between 2 different genes, you could say that they are 28 map units apart. Okay? Think about that. All right. If you get a 28% recombination frequency, you can say that the two alleles, two genes, are 28 map units apart on a given chromosome. And important is that the map units indicate a relative distance, not a physical distance measured with a ruler. So just keep that in mind. Okay, so let's start with this deal. We've got a sperm and an egg. You've got the parental generation gametes to produce the F1 cell here. All right, the F1 cell is diploid with 6 chromosomes, and it's heterozygous for all 6 of those genes. The red chromosomes represent moms. The blue represent dads. Now, each of these chromosomes has hundreds or even thousands of genes, but we're only going to concentrate on 4 specifically on the longest chromosomes and 1 each on the middle-sized and shortest chromosomes. Now, using genes E and D, the alleles of unlinked genes are either on separate chromosomes like D and E, or they're so far apart on a long chromosome like C and F that they assort independently. And so genes and genes on the same chromosomes are so very close together do not assort independently, and they are said to be genetically linked. So these guys, the ones that we're talking about up here, have what we call a 50% recombination frequency. All right, because they're assorted independently. These guys, the ones that are really, really close, they would have a much smaller recombination frequency. Now, we can use the recombination frequencies to map out the distance between 2 chromosomes. So, for example, if there is a 9% recombination frequency between gene B and gene CN, which is the black body gene, and I'm not sure what the CN was. And then there's a 9.5% recombination frequency between that CN gene and the vestigial gene, then we could expect to find 9 map units between B and CN and 9.5 map units between CN and VG, or vestigial, which means that we could also expect to find a 17% difference between, or recombination frequency between the black-bodied gene and the vestigial gene. If you go back to the data, here, look at what we get as a recombination frequency, 17%. All right. So, and this is what Morgan actually found. All right. So he was able to predict map frequencies using recombination frequencies. Okay. So genes that are far apart on the same chromosome can have a recombination frequency near 50%. These genes are physically linked, but genetically unlinked because they have such a high crossing over rate. So they behave as if they're found on different chromosomes. And this is just a map that a group of scientists working with Morgan were able to work out, finding the various things. And these are all traits that we find on a chromosome in the Drosophila genome. Okay, so that is it for now, my friends. I will see you, I suppose, on tomorrow. All right, have yourselves a lovely day.